Swayam Prabha. Digital India. Educated India.
Continuation with the cell signaling. So, the one next important step is find the signal ligand bound receptor transduction, then gene expression is altered, right. So, targets are either activated or suppressed. And now we also saw that this regulation is very fine control. The moment you do not have signal, then right away it stops. There are situations where the activated state needs to be maintained, okay, so without continuous signaling, because that particular cell type, let us say a particular differentiated state is marked by expression of a certain set of genes. Then if those expressions are not going to happen, then you will not have that differentiated state. So, how do you maintain that state of transcription, either on or off? So, that we are going to look at in the next couple of slides. So, there are 4 different mechanisms by which this happens. In one situation, once activated, that particular transcription factor may bind to another enhancer and continue to activate the transcription. Okay, the product itself is an activator. Okay, so there could be multiple targets activated. One of them may be an activator. Okay, that product itself goes and binds via another enhancer, continues to activate that gene. Right. Yeah. So that's one example like that. So where it's slightly different here. Well, I would not say it is slightly different because that also has modules, right. So, here I am seeing 2 different enhancer, initially activation may be via one and then it works via the other one once this product is produced. And the second one is a permanent chromatin modification, permanent meaning during that particular organism's lifetime. In that particular differentiated tissue, you may have these proteins like trithorax complex proteins. They bind and prevent nucleosomes from assembling there. So, they maintain euchromatin status. Okay. So, this is another mechanism. And a third one is one of the downstream activated target may be coding for the ligand, same ligand. So, therefore, it keeps cycling. And a small variation of that is the ligand is produced by an adjacent cell, okay, so that it continues to do this signaling. At least I know in C. elegans germ cell niche, the somatic cell that produces the delta ligand and the germ cell that produces the notch receptor, they need to continuously be doing this. If you get rid of the niche cell, meaning you get rid of delta, then these cells will right away differentiate, they will not remain in the mitotic potential. So, there are situations where the adjacent cell producing ligand continues and that is required. So, these are 4 different models to explain how the altered state of transcription, I am carefully using the word altered because it's not always activation or inactivation. So, in one, as a consequence of one particular signaling, you may have both. Some genes are activated while other genes are inactivated. So, that altered state, how that may be maintained, these are the models to explain that. Okay. So, the next one we are going to look at the ECM, extracellular matrix. Okay. First, let us familiarize with what is ECM and what are its components, and then look at how it affects development. So, this is an in an embryo you are seeing ECM, a continuous, the fibronectin, a particular component of the ECM expressed, and that is absolutely important for the continued embryonic development. Okay. So, we will see details of that. So, this figure B shows you the ECM. So, this area you have the epithelial cells, and below this mesh like structures is the mesenchymal cell there. Okay. So, the epithelium produces an ECM which is made of a protein called a laminin, and that form a mat like mesh, densely woven sort of mat like mesh, this sheet that you are seeing where it is labeled basal lamina. So, that is the ECM secreted by the epithelial cells, okay, primarily made of laminin. And below that you have the ECM coming from below the mesenchymal cells, that is this mesh like structures, okay, having collagen, fibronectin, then proteoglycans, primarily heparan sulfate and chondroitin sulfate. So, these form the mesh like structure. Okay. So, they are the components of the ECM. ECM is primarily collagen is a major protein, the other one is fibronectin. Fibronectin helps in connecting from the cytoskeleton all the way to collagen and the carbohydrates. So, let us say the main structural component in ECM, the protein component is collagen. Then in addition you have proteoglycans, where it is a protein carbohydrate combination, but the carbohydrate portion is large in compared to the protein part. If it is opposite, you call that as glycoproteins. Okay. So, they are also present, but the proteoglycans are the main components, and this is the composition of the ECM. But the ECM is connected, like for example collagen or proteoglycans would be bound covalently by fibronectin, that would in turn interact with another protein called integrin, which is a transmembrane protein, and these integrins connect to cytoskeletal component, primarily at the periphery of the cell just below the plasma membrane. What kind of cytoskeletal material you have? So, you have a mesh like network of microfilaments. So, that is what integrin connects to. So, we will see that in the next one, but before we go there, so we want to know these 3 points. ECM is a source of signaling for development. ECM is critical, and some of the ECM components, like for example proteoglycans, they have very high affinity for the signaling ligands like Wnt and so on. So, they bind and they help in getting the ligands concentrated for signaling via the plasma membrane bound receptors. So, it is not as though ligand is directly going to the plasma membrane. Okay. So, in between you have an ECM, and there are ECM components that help in facilitating this interaction, ligand to receptor, and sometimes ECM itself is a source. Remember the permissive induction we learnt, instructive induction and permissive induction. Instructive means the inducer tells, these are the instructions for you to execute this, and permissive means a neutral conducive environment that itself enables the responding cells to develop in a certain way. So, the permissive induction is ECM is one example of that. Okay. And I told you already, important for facilitating the paracrine ligands like Wnt, I told you as an example, FGF another one, and ECM is critical for cell attachment, migration, they kind of guide which way to go. So, migration is important, direction for migration is important, and differentiation as well. We will see an example of how differentiation happens in the presence and absence of ECM, and morphogenesis as a consequence of the differentiation. So this is a cartoon view of whatever I first said. So you have this fibronectin, okay, with its arginine, glycine, aspartic acid repeat units that act as a place for these integrins to bind. So this is a beta integrin that goes as a continuous chain all through the membrane, and then you have alpha integrin where you have this disulfide bond connecting these two. So this is the kind of thing that is there in notch. Remember I told you notch first gets cleaved even before going to the membrane, and therefore it is a heterodimer, and this is how that would look like. Okay. So this is integrin, alpha and beta integrin. So they connect to fibronectin, and fibronectin in turn connects to collagen and proteoglycans, etc. And these integrins inside the cell link to the actin microfilaments. See, in this cartoon you are just seeing two short fiber like thing, but in reality it is a mesh under the plasma membrane, very much looking like this but below the plasma membrane. So there again you have this vinculin, talin and actinin as intermediates in that interaction between integrins and the actin microfilaments. So this is how it is all linked. Okay, the cell internal cytoskeleton, like the structural components, are linked to the ECM, which again links similar to another cell. Right? You need to imagine this whole thing, another one existing on top here. So on the other side. So that is the ECM. So now let us look at its developmental consequences. So here you have these rat Sertoli cells. The Sertoli cells are important in where? Anyone have heard this term? Haven't heard of? Leydig cells and Sertoli cells? Yes. Yes. So these Sertoli cells are important for germ cells to proliferate in testes, in mammals. So when you grow them, these isolated Sertoli cells in a tissue culture plate, plain tissue culture plate, they just grow like a layer. But if it is coated with the right ECM, it differentiates like this. So they provide the niche where the germ cells differentiate. So without Sertoli cells you won't have the germ cells formed in the testis. So you see the difference. So these are grown for same amount of time and taken under similar magnification. So this is the impact of presence and absence of ECM. And this is explained by a cartoon in mammary gland. So in mammary gland again, see, glands are mostly tubular structure in which cells in the cortex produce and secrete whatever they are secreting to the tube in the middle. Then production from multiple such structures are collected into one particular place. So mammary gland development again happens similarly. So this is the cells, epithelium like cells. These are the ones that are going to finally form the mammary gland tubes, but without basal lamina. Remember, basal lamina is this, right, formed of laminin secreted by epithelium, and without that they just grow like that, and they will continuously they will be producing proteins involved in cell proliferation like c-Myc and cyclin D1, and the differentiated, the casein production, whey protein production, so they all will not be produced. So these are all the products of mammary gland, differentiated mammary gland, so they are not going to be produced. But once you provide the ECM and you have this integrin attachment, okay, so when that attachment happens, remember integrin is transmembrane on these mammary gland cells, right, and they attach to the ECM. Where do they attach to? Fibronectin, yes. So when that attachment happens, these cells kind of now form a lump like structure instead of sheet, and then they wrap themselves by drawing this basal lamina. They produce the basal lamina themselves, remember, and that induces differentiation, and as a mark of that you will see lactoferrin coming on and this P21 transcription factor coming on, but still these are yet to form, but then these proliferation genes are now off. So the differentiation has set in. Then they form the basal lamina all around, so the tube is formed, and to the lumen these cells now they are differentiated and they will produce the casein and lactoferrin, and then they stop these, but the whey protein is not yet produced. In the next level even that is going to be produced. All the milk components will be produced and the proliferation is now stopped, and then you form the tight junctions as well. So therefore the luminal content does not leak. So that is the context in which you see the tight junctions. So another place where you see is the intestinal mucosa. So there again you have tight junction. Tight junction means you have the lipid bilayer fusion. So there are no protein based junction. So those are like desmosomes anchoring a cell to adjacent cell or to the basement membrane, or you have gap junctions where it is like a pore where things can freely move from one cytoplasm to another cytoplasm. So as opposed to those two junctions, this is a membrane fusion. So therefore there will be no leakage. Imagine the food that you are eating leaks between this interstitial space of the intestinal mucosa, you will just die, okay, due to all the reactions that will start. So that happens. So now you have a milk secreting mammary gland tube. So you saw the role of basal lamina. So here we are not doing a genetic experiment, therefore you did not see, only in a cartoon version you are seeing where these attachments happen. But if you break down this attachment, then this will not happen. This differentiation happens only after this attachment. So that is why ECM is critical. Alright, then we get into the next one where this epithelium to mesenchymal transition is crucial in different situations, but widely popular and appreciated in the context of metastasis. Okay. So you have epithelial cells having this, the desmosome connection anchorage, and then you have cadagrine, then you have the catenins and then actin. So right, so this adherent junctions. Now and this one now undergoes changes when you are going to have epithelial to mesenchymal transition. The first thing is because of the signals, the cadagrine production goes down and the catenin and cytoskeletal element like actin, they rearrange, then the basement membrane gets dissolved and then this comes out, and now it has become mesenchyme. Then this can go wherever it wants. So this is how an epithelium based origin cancer become metastatic. But this is not like totally an unusual pathogenic situation. This is basically a normal process having gone wrong in a wrong place and time. But normally this is required to make these neural crust cells to come out from that epithelium like structure to migrate to other places to make other kinds of cells. Remember, the pigment forming cells come from there, they migrate away. So they come out like this. So this EMT is critical during embryonic development. So similarly this mesenchyme that comes from epiplast, so you will know what is epiplast, hypoplast, etc. when we learn mammalian embryo development. Okay, so when we go to that we will discuss that in detail. So essentially you have a sheet like cells from which other types of cells need to form in the early embryo, and those cells that come out to make the mesoderm, they require the CMT process. Okay. So these are the really crucial stages in embryonic development where EMT is absolutely essential. And only when this process happens in the wrong place and time you get cancer, cancer metastasis. So cancer origin is different from this metastasis. So this is EMT transition. So now we move on to the next important concept in developmental biology, that is cell differentiation. So here again we will go to some interesting history as we go along. Okay. So first, so we are now actually from an organism or a tissue perspective, we were mostly talking about morphogenesis, which is at a tissue level. So now before that you need to have cells trying to become different kinds of cells, and that process we call as differentiation. So a differentiated cell, like for example mammary gland, functional mammary gland cell, of course will have its own unique biochemistry and morphology that you will be able to identify it as a mammary gland cell. But that is a very late phenotype. But long before that there are things that change where the cells still look very similar, but then they have already made up their mind. Okay. Like you could be thinking, one of you here could be thinking, I am going to go for lunch right after this, then someone will be thinking I am going to go to lab, someone will be thinking I am going to go to our next class. But we won't know all of this now, right? But these ideas are formed in your mind. So that is what is commitment. Okay. So commitment. So the commitment itself has different stages, a progressive set of situation. So two easily identifiable states are specification and determination. Okay. So differentiation is end process of all of this. So specification is the cell is now induced, it knows what it wants to do, and it is ready to do as long as no one is going to give an opposite signal. So if you take this cell and put it in a neutral environment where no one is giving an opposite instruction, it will go on to, let us say, mammary gland, okay, mammary gland cell. So that stage is called specification. So the key thing here is the required molecular changes have happened for a particular development, like to become mammary gland, but it can change its mind. So if you give an opposite signal, you can still reverse this process. At that stage you call as specification, where reversibility is still possible. And if you have gone couple of more steps in the process of developing into that particular type, where regardless of any new instruction, now it is not going to listen, it is too late, it's gone beyond the stage of coming back, and that once a cell reaches that stage, then you say it is determined. First it is specified, so at a specified state it can still go back if the right instructions come, and when it has reached a determined stage, then it is irreversible. So these two are the subclass of commitment. But even after determination, the cells might still not look like a fully differentiated one morphologically or biochemically. They may not still be producing casein. Casein you are not going to see in other cells, you will only see in mammary gland cells. Like just to give you an idea of what is a biochemical difference or morphological difference. Okay, so they may not have happened yet, but all are set already for that. So now we are going to look at specification, like there are different types of specifications. So we are going to look at each one of them and where they play an important role. So we go back in history. So before I get into this, I should show how this organism looks like. Yeah, this is how they look like. Okay, these are mollusks, Patella. So now in these mollusks, we are going to look at how during embryonic development specification happens. So in these very early stage, if you go and isolate individual cells, so essentially you can put them in calcium free water and shake, the cells will loosen up, and that is why these kind of organisms. And when you take them and you see again you have different color, these are, although it is cartoon showing a color, but these cells do have pigments that are useful in distinguishing the different kinds. Now you take them and each cell makes a part of what that embryo will be. So as part of this embryo, whatever structure this particular cell would have made eventually, that structure it makes even when isolated. Okay, so this divides into 2 and then 4 and then goes on to make these structures, whether it is on the embryo or outside. It does not make the other blue color cells or the blue color structures. So each cell seems to have instruction to do what it is supposed to do, and it will do it regardless of the neighbors, and that we call as autonomous specification. So the cell is autonomous now, and as a result the final embryo is viewed as a mosaic. You have the different structures all put together with no connections to each other. So that is why we call this as mosaic development. So organisms where you have autonomous specification, the development is going to be mosaic development. So the opposite also will be true. In this embryo, if you take this out, then the rest of the embryo will make a final structure missing part of this, what would have come from this cell. Okay, the opposite also true. So that is why it is mosaic. So you remove one tile on the mosaic floor, only that tile is missing, the rest of the floor is not partially compromised, they are totally fine. So this is autonomous specification. So Chabry did an interesting experiment with these kind of organisms. So this is tunicates, okay, tunicate, here you have an example, another one more. Since you do not go to beach and you do not look at stuff, so I will bring images. So these are filter feeders, they allow water to come and filter out the water and the food is staying. So that is how they live their lives. So their embryos again easy to separate the blastomeres, and you can do this fate mapping. So you already know what these colors mean, right? So what this color means? B4.2, it is going to make ectoderm, like the final ectoderm comes from at 8 cell stage, it comes from this, probably from the next one also, and this, right. So what this means is composite color. So this mesoderm, like the muscle mesoderm, mesenchyme, endoderm, all are going to come descend from this one cell. That is what the fate map means. So now you go and separate these cells, okay, and they make the respective parts as we just understood in autonomous specification. Right, these are going to make only the ectoderm, and this is going to make these structures based on this fate map, and this is going to make these structures based on this fate map. So the different parts separately made, mosaic. If you put them all together, then you have a complete embryo. So Chabry did not stop there, he tried to experiment, right, observation, the next experimentation. So what he did is he pressed at 4 cell stage with a needle on these 2 ventral cells such that some amount of the cytoplasm, when the spindle forms, gets into this animal pole derived cells. Okay, so they end up becoming like this. Now from these descendants you have muscle forming cells coming out. Okay, this is consistent, this told, yes, instructions are there, and if you put the, and the instructions have already gone separately. So everything came as maternal dowry in the oocyte cytoplasm, and now obviously at the very starting these components, the maternal determinants, okay, remember this word, are asymmetrically distributed in the one cell embryo, right. So then only this is going to happen. And now when I mix that asymmetric distribution, then new structures can come. So whatever would have gone into this, now I am trying to push it into this, and that is going to, that determinant is going to induce this cell now to give descendants to make muscle cells as well. So presumably the next division or next subsequent divisions, some cells got only the determinants to make the ectoderm and they made ectoderm cells, and some instructions that is going to make muscle, probably some descendants of this B4.2 had only that and they ended up making muscles. That is what this means, tissue including muscle cells. So this is an experiment that is consistent with the idea that you have determinants all coming from the oocytes, and now which cell inherits which instruction, that is going to make that structure. Continuation of this, we revisit this Weismann's germ plasm theory. We have already learnt this, right, genome equivalence, remember. The difference here is that what August Weismann proposed is, it is all on chromosomes and the entire set of chromosomes stay with the germ lineage, okay. But the other cells that come, they get parts of this chromosome, okay. Instead of imagining the cytoplasmic component, here you are assuming chromosome, but each one gets one different part of the chromosome, therefore you have differentiation. And he, as an extension of this, he proposed, if you take a frog embryo, cut the animal pole and vegetable pole separate, then they will form only half embryos, right, they will not have the other instruction. So Roux wanted to test this out. So he did an experiment where he killed half of the embryo with a hot needle. So half dead embryo. So then when it completed embryonic development, you had half embryo and the other half was missing. Perfect, okay. Autonomous specification, mosaic development. So the previous people are all correct, okay. Chabry and August Weismann, they are all correct. They are correct, only thing is partly, not completely. So there is a defect in this experiment. Can you figure out what is the defect in this experiment? So we will revisit this experiment a little later after doing some other experiment. So here enter Driesch. So what he did is, he took the sea urchin, the place where he lived, the coastal area of France where sea urchins were abundant, so he experimented with them. So he took the embryos and then separated them into individual cells, and to his surprise he ended up finding each one of them developed into a complete larval stage. So as if each one had the information to make the other missing parts. So this is already shaking the autonomous specification. So he could have stopped at this level, but he went ahead to do some experiment that changed his life, okay. So what he did is, so this is the normal cleavage pattern. So it is going to divide, so this is a cross section view and this is the top view or side view where you are able to see both top and bottom. These are the cell division patterns, like the number tells you what came from what. And then 16 cell stage, it will look like cross section, and surface view it will look like this, and so on. So he did something like what Chabry did. He tried to mix the division planes by pressing them between two glass plates, and when he did that, the spindle orientation changed, and as a result these kind of mixed thing happened, okay. So you look at the color codes here and here, 8 cell stage, and compare with this neat pattern. So now you should have a totally mixed larva, right, probably the tail on the head and eye at the end of the posterior part and so on. Didn't happen, formed a normal embryo. So that made him to come to these conclusions. So a particular blastomere, whatever its structure it makes in the embryo, that is not only its capacity, it has additional capacities. Go back to vulva, okay, tertiary fate, Any one of the 6 can be in that, any one of the 6 can be secondary, any one of the 6 can be primary. But the ones that do not get those instructions, they automatically become tertiary, but if you remove the negative signaling, then they will all become primary too, right. So that is the conclusion he came to. A blastomere, its developmental potential is greater than what it normally does as part of the embryo. And if that is so, then why does it not make all the embryonic structures in the embryo? His obvious conclusion for that is the neighboring cells must be telling it, okay, you make only this, do not make anything else. And the third one, which means then a cell, whatever structure it makes, why does it make that structure and not other structure? It depends on where in the embryo it is and what instruction it is getting. So these are the 3 conclusions he came to. So obviously you are seeing genomic equivalence here. So there is nuclear equivalence, otherwise how a cell had all the potential at the early embryonic stage, and at the same time cells having the same genetic material, how they are able to interact. Okay, the cell induction and response and the genomic equivalence made him to think that, okay, this is not in the realm of science. Okay, so probably there are other things other than science. So there is some force that instructs the embryo to do something, and that is something I can't figure out. He just thought that the embryo develops like the way you make a machine. So in a machine making context, can you think of this playing out? Like I forgot to bring couple of gaskets in joining some pipes. Now will I be able to join the pipe by converting something else in transforming into gasket? It is not going to happen. So he was dejected and depressed in life and quit science and moved on to become a saint or something like that. Okay. So his experiment had such strong consequences to embryology as well as personal life. So such is the mindset of true scientists. So that is the story of Driesch. But remember, whether you remember his personal life consequence or not, these 3 you need to remember. The developmental potential of a given blastomere is lot more than what it actually does in an embryonic context, normal location. Which means the neighbors must be telling it not to make the other structures. Which means whatever structure it makes is probably due to the position on the embryo. Okay, now we will revisit the Roos mistakes. So he did the destroy and see experiment, but he did not do the remove or isolate experiments, and that is done here. So you take a tissue here and put it elsewhere, and it is not going to make brain somewhere near the belly. Okay, so instead it makes belly only there. And in an opposite experiment, although not shown by this arrow, this is not going to make a part of intestine on part of brain. Instead it is or the epidermis, skin epidermis, it makes proper skin epidermis. Okay. So this sort of thing we call conditional specification, which is leading to regulative development. The neighbors regulate. So the condition is where you are and what conditions are put on you, you being blastomere by the neighboring blastomeres. So that is the conditional specification, and that leads to regulative development. Autonomous specification, mosaic development. Conditional specification, regulative development. Okay, clear? So you see the kind of beautiful experiments and you see the profound conclusions. So if you remove it, is not going to miss that part, unlike what we saw in that mollusk. Here it is going to form a normal thing. Okay, the last thing is syncytial development. This is not very common, it is only in some insect species. Here the cartoon is from Drosophila, that is the insect that is most studied. So here essentially in the early embryonic development you have lot of nuclear divisions without cytoplasm being separated, no cytokinesis. So the embryo becomes a sac containing lot of nuclei, but the cytoplasm is asymmetric, meaning the components of the cytoplasm in different parts have different components. So color coded here, see the red in the anterior, meaning some component is present in the anterior, not in the posterior, and vice versa. So what a particular nucleus is going to develop depends on where in this concentration gradient of the asymmetrically distributed molecules, and this we call as syncytial development. So it's the embryo is a syncytium. Syncytium definition is multiple nuclei in a single cell, single plasma membrane bound cytoplasm, that is syncytium. So this is a syncytium, the early embryo of Drosophila. So you have rapid synchronous nuclear cleavage divisions with no cytoplasm being separated, and the position of the nucleus in that cytoplasm determines what it is going to become. Right. So here is an example of anterior determinant called bicoid. We have encountered this, right. It is a transcription factor as well as a translation factor. So in what avatar did we encounter it? Prevents translation. Right. So we saw it right after seeing masking, okay, as an additional example of how translation suppression works, right. Bicoid. So it acts as a transcription factor as well. Anterior specifying molecules are induced by bicoid. And caudal, so you just told caudal mRNA is translationally repressed by bicoid. So therefore here caudal is not produced, it is produced here. Okay, and this we will see in detail when we are going to learn about Drosophila embryogenesis. Since a whole lot of signaling pathways and an idea, a paradigm for embryonic development comes from studying Drosophila embryonic development. So we will go through that in detail later, not right now. So this is a key experiment. Okay, so this is so you should actually read about her biography and get inspired. Okay, Naslin Walhard, because I can't recollect this enough to be saturated. So these experiments that she did were not like long ago in history. These are all in mid 80s. So she did simple experiments. All she did is mutagenize and screen for mutants where the embryos are dead, okay, and then look at why the embryo is dead. No ultracentrifuge, no complex protein purifying equipments, none of those, no cDNA library construction or northern, southern, western, etc., none of that. She did simple mutagenesis and look at what's wrong. So one of the mutants that she got did not make the head structure. So this is the wild type, okay. So this is syncytial state. So you have normal development, you have the head and the tail and the segments, thoracic segments like the chest part and the abdomen segments. In the bicoid mutants, no anterior structure, instead a tail like structure forming and then central abdomen. So she just took some cytoplasm in the anterior of wild type egg, put it into the bicoid mutant, and you got anterior structure, okay, and put it in the center, formed the head in the middle and two tails. So she proposed bicoid problem makes a concentration gradient, and that determines highest concentration means head, lowest means tail, and wherever you put the highest concentration you are going to hit the head, okay. So this and similar analysis using genetics is how we learnt all those pathways that we were learning, and not surprisingly she got Nobel Prize, okay, eventually for this embryonic development.